Okay, so in this lesson we're going to go over the workings of the cell. Okay, we'll talk kind of in depth about what the job of each organelle is. Okay, but we're going to do that in the context of how they work together. Okay, what processes they use in order to help the cell to function properly. We probably won't get to the stuff on cell transport today. Okay, but that'll be an important thing that we'll go over tomorrow. Okay, so my plan is kind of get through all the functioning of the cell stuff today, talk about cell transport tomorrow, have some time to do a pre-lab, Friday go into the lab and look through the microscope. Okay, so that's kind of the plan. Okay, because remember it's already Wednesday. If you looked at me strangely like I was going crazy, remember it's already Wednesday.
Yeah, but we didn't.
And we didn't have to listen to Celine Dion this morning. So that's awesome. Okay. The microscopy lab's due Friday. Yeah. Okay. Oh, actually, sorry. Okay. So number one. Okay. First key point. Understand the significance of compartmentalization. Okay. This is going to be an important term. In fact, today we're going to go over lots of big words. Okay. Lots of important vocabulary in biology. Okay. Especially to do with the parts of the cell. Now, yesterday at our PD day, this was one of the things that came up in the science department, is that people have this idea that biology is I just have to read and memorize and regurgitate, okay? But it's very much not that way. Yes, there are a lot of facts to know in biology. Yes, there's a lot of stuff to memorize in biology, but we're not testing your memorization skills, okay? We want to know, can you apply that stuff? Okay, so when we ask you questions on a test, yes, there's going to be some stuff that's recall. Like, okay, here's a diagram of a cell. Tell me what the parts do. Yeah, there'll be some of that. Okay, but more importantly, there's going to be stuff about, here's a cell in a situation like this. Using what you know of a cell's functions and anatomy, what might the cell's responses to this situation be? Okay, now that's saying, okay, I know that the cell membrane does this, and I know that this process allows certain things to get in through the cell, and it seems like in this situation, maybe one of those things isn't going to function correctly, and that's going to have this effect on the cell. So it's not something you can memorize. It's something you're going to have to take what you know and apply to that situation. Does everyone kind of understand the difference? So there's a lot of application in biology. There's also a lot of creation in biology, and that is come up with a situation where this process may not work, okay? And you would have to think, okay, how does that process work? Because I memorized how that process works. Now, how does that work here? Okay? Or I give you a situation and ask you to evaluate what's going on in that situation. You still have to have the knowledge to do that, okay? But it's a much higher level thinking type of question than what does the mitochondria do? Okay? Everyone kind of follow me there? So when you're studying for biology, certainly there is a certain amount of memorization, but that's not the important part. Okay? When you're studying for biology, think about all the different ways what you're learning can be applied to something. Right? And we'll go over some questions like that so you kind of get an idea, but that's the big part of biology. So don't think it's just memorize and regurgitate. It's not. It's so much more than that. Okay, so compartmentalization. What is a compartment? An area, a room, okay? Yeah, something that's closed off from everywhere else, right? Okay, so a cell is compartmentalized because it's got organelles, okay? Each one of those organelles is a separate compartment within the cell. That's what compartmentalization is. There are basically two kinds of cells. Now, I don't mean plant and animal. I mean, there are prokaryotic cells, like bacteria, that don't have organelles. They don't have compartments. And then there are eukaryotic cells, like we have. They do have organelles. They do have compartments. The implications for that are fairly widespread, and that's what we'll talk about. Okay? Know the role of each organelle. Yep, that'll be on tomorrow's quiz. So know that. Okay? Understand that cells are a complex system of interdependent processes. Okay. What does interdependent mean? Right, they're dependent on each other, and not necessarily in a 1, 2, 3, 4, 5 type of an order either. Okay, if process 3 stops working, that could affect not just processes 4 and 5, but it could also affect process 2, okay, or process 1. They're interdependent. It's like a web as opposed to a chain. Okay? All the processes within the cell are dependent on each other. Okay? All right, understand how a cell carries out its functions. That's kind of what are the functions of the parts of the cell. Okay, and then number 5, understand how materials are transported. We said we probably won't get to that today, but probably tomorrow. All right, compartmentalization. So compartments within the cell. Cells are divided into compartments by membranes. Each organelle has its own membrane that separates it from the rest of the cell. Okay, so that's what compartmentalization is, and it is present only in eukaryotic cells. Okay? All multicellular organisms are eukaryotes. Okay? They have eukaryotic cells. Right? Some single-celled organisms are eukaryotes. The ones we looked at the other day are all eukaryotes. The amoeba, the paramecium, and the euglena. Okay? They're eukaryotes because they have all those internal structures. A bacteria is a prokaryote. Okay? A bacterial cell has basically none, or has basically no membrane-enclosed organelles. That means all of the processes that a cell has to carry out, because point number 2 of the cell theory is cells carry out basic functions. Whether you're prokaryotic or eukaryotic, that's true. If you're prokaryotic, you have to carry out all those processes in the same room. If you're eukaryotic, you can have a separate compartment for each one of those processes. That is more efficient. That allows those processes to happen more effectively. Because that compartment will have exactly the conditions for that process to occur. If you're a prokaryote, the internal conditions of your cell are the same for everything. Right? And that means that maybe those aren't ideal. They're good enough for everything to work, but they're not ideal for anything. Okay? It's like if you've got 10 different people and you're only buying one pizza. Okay? You're going to get a pizza that everyone's okay with, but nobody loves, unless you're that lucky person that actually does. But okay, and for all intents and purposes, you're not going to be able to satisfy everybody. Okay? It's also similar to your house. Okay, and that's the example I have in here. Within your house, you have 3 separate temperature environments, maybe 4. Okay, if you have like another type of fridge, but okay, you probably have 3. You have the part of your house that you live in, and you keep that between 20 and 22 degrees. Okay, that's comfortable for you. Okay, that's where the human body kind of is most comfortable. Then you have your refrigerator. You keep your refrigerator around 4 degrees Celsius, okay? Because that keeps the food in it from spoiling, okay? Your food lasts longer at 4 degrees Celsius than it would at 20. And then you have your freezer, which is for long-term storage of food, okay? It's at about minus 15 to minus 18 degrees Celsius, okay? That's what a eukaryotic cell is like. It has different environments for each process. A prokaryotic cell would be like a house with no refrigerator and no freezer that's kept at 10 degrees Celsius to try and find a happy medium for all 3 processes. Are all 3 processes working very well? No, you have no ability to freeze anything, so you cannot store anything really long term. Okay? You're certainly not keeping stuff fresh as long at 10 degrees as you are at 4 degrees, and you aren't very comfortable at 10 degrees Celsius. You have to wear a coat around, okay, all the time in your house. Okay? Not ideal. Right? Now, I don't want you to think that that means all prokaryotes are at a disadvantage. It doesn't mean that, actually. Okay? There are advantages and disadvantages to being eukaryotic versus prokaryotic. Eukaryotes, yep, they can get really big. They can become multicellular because they can have these more efficient processes going on. They can also become specialized. The cell needs more mitochondria, it can do that. Okay? If it needs more cytoskeleton, it can do that. A prokaryote can't. It doesn't have those things. So you don't ever see a multicellular prokaryote. But by the same reasoning, because a prokaryote can withstand and tolerate conditions that may not be ideal, we often find prokaryotes living in places where no eukaryote would ever survive. Okay? We find them at the bottom of the deepest, darkest parts of the ocean. We find them in the hottest parts of a hot spring in Yellowstone National Park. Okay? No eukaryote would ever be able to survive in conditions like that, but they can. Okay? So it gives them a bit more survivability. okay, versus a eukaryote, or a wider area. Okay, compartmentalization makes sense to everybody? I usually ask something about compartmentalization when we do an exam. Okay. All right, so compartmentalization of cells allows cells to become larger. Okay, prokaryotes have no compartments and are much smaller than eukaryotes. Okay, and eukaryotes are more specialized in their function. Okay, so a bacteria looks like this. This little snarly thing that you can see kind of in the middle, that's its genetic material. No nucleus. Okay, it's in there with everything else. So the cell is digesting its food, processing its wastes in the same place it stores its most valuable material. Okay, that would be like you putting a gold bar in your toilet and eating there. Don't eat it on the toilet. Okay? Okay, but that's essentially what a prokaryote does. All the processes have to happen in the same place. Okay, a eukaryote, it's got a bathroom. That's where that goes on. Got a kitchen. That's where you make your food. A dining room where you eat it. Maybe you don't have to separate one, but whatever. Okay, different places for everything. All right. So DNA. What's the purpose of DNA? DNA is used to make stuff. Well, it at least tells us how to make it. Okay, it's the instructions. Right? So in the environment of the nucleus, so basically this is telling us here's what the nucleus and the DNA stored within it do. Okay? So within the environment of the nucleus, DNA is stored. Okay? We call the DNA chromatin because it's kind of all unwound. You guys have probably seen pictures of what chromosomes look like. Okay? They only look like the pictures of them when a cell is dividing and all the DNA is condensed into those forms. Most of the time, the DNA is completely unraveled. Okay, so what you see is like a ball of yarn. Okay, the picture of a chromosome is like a ball of yarn. Most of the time in the nucleus, that ball of yarn is completely unraveled, mashed into a mixture, just packaged. Okay, so your nucleolus has got to know where everything is, and it's got to look through a ball of yarn for a particular section of the yarn each and every time. Okay, it's amazing anything ever gets done. Okay, so the nucleus stores DNA. That's really all we need to know. Okay, looks like this under an electron microscope, a scanning electron microscope. It looks like this on the surface. Okay, these are the nuclear pores here that you can see. Okay, and this is even more. This is the surface of the membrane. You can see. I told you it kind of looked woven. Okay. All right. Questions on the nucleus? All right. This is the process of protein synthesis. Again, it's not something I'm going to test you on, but I told you how it works. Okay, I'm never going to ask you again. It's just people always want to know. Okay, so what happens is inside your nucleus, okay, is your DNA. That's the blue double-stranded stuff up top here. Right? Your nucleolus has an enzyme in it called RNA polymerase. You don't need to know that either. Okay, all that does is pull the DNA apart and allow it to be copied. Okay. RNA polymerase basically makes RNA by copying one side of your DNA. Okay, so it copies the DNA and it sends that copy out through a nuclear pore, okay, which you can see right here, right, out, and it merges with a ribosome, and the ribosome is like a foreman. Okay, so it's a foreman at a construction site. It knows where the raw materials are, it knows whose job is whose, and it kind of plans everything and gets the job done. Okay, supervises the job. Okay, so it will help with the production of proteins. Okay, that's protein synthesis. Nucleus copies DNA, RNA goes to the ribosome makes the proteins. And that's the extent to which you need to understand it. That's probably even beyond for Science 10, the extent to which you need to understand protein synthesis. Okay, so the nucleolus produces RNA, okay, which is a copy of the cell's DNA, okay, and that is used to help make proteins. All right. So kind of also within this, we learn that the ribosome's job is to make proteins. Okay, so the ribosomes make protein. Okay. Ribosomes, proteins are created. Yeah. Okay. Questions on that one?
So where's the RNA stored?
RNA isn't stored. It's produced. It's a copy of your DNA, and it's sent out into the cell. Okay, so like the cell obviously doesn't want to cut your DNA and go, here's the most valuable piece of information I have about making this stuff. Don't use it. Okay? Realistically, first off, you don't want to do that. And secondly, you're going to have more than one ribosome doing that job. You're going to need more than one copy. Okay? So that's why it all stays in there, right? It's not a lending library where you actually sign out the real book. It's like, okay, that's the real book. I'll make a copy of it for you. And if you lose it, no big deal.
So does the nucleolus controls the ribosomes, or like tells them what to do?
It doesn't really control the ribosomes. It just sends the information to them. Okay, now endoplasmic reticulum. Okay, we said that this stuff was fairly important here. So much of the volume of the cell is filled with this stuff, and I was telling you that it doesn't really show on a diagram just how much ER there really is within the cell, because it would just fill it right up. So within the membranes of the smooth ER, the following things are done. Okay, so smooth ER. Lipids are synthesized for use in building or repairing membranes. Complex carbohydrates are broken down. Drugs and poisons are detoxified. Okay? Another thing that can happen in both types of ER is hormone production. Now, most of the time, hormones are made by the ribosomes because most hormones are proteins. There are some that are fat-based. If they are fat-based, they are made in the place that makes fats. That would be the ER. Okay? Now, on a test, I would expect you to write one of those functions. Okay, I'm not going to ask you for all 4 functions of the ER. One would be good. Okay, and this picture down below is actually another electron microscope picture of the ER. So this stuff here on this side, this is the rough ER. So it looks kind of organized. Okay, it's layered, and the little bumps, the little black dots, those are all ribosomes. Okay, over here, it's all chaotic, and there are virtually no ribosomes. That's the smooth ER. So the smooth ER is on the left, and the rough ER is on the right. Okay, they look very different under an electron microscope. Okay, questions on the ER? All right, now lipids, they need to be moved quickly to the cell membrane, so the smooth ER is also in contact with many parts of the cell membrane. Okay, so it's basically attached to it, okay, in most places. We also want to limit how much any of those detoxified poisons are touching any other parts of the cell. So there's another reason why the smooth ER is attached to the cell membrane. It means that any detoxified poisons can be excreted right away without having to go to the Golgi apparatus, without having to go anywhere else and possibly damage other parts of the cell. Okay, the Golgi apparatus, okay, like we said, kind of like a garbage man of the cell. Now I was telling you, there's a receiving side and a shipping side, okay, the cis face and the trans face to the Golgi apparatus. When they draw a diagram of it, like draw this picture, it doesn't really show that very well, okay? But when you see it under an electron microscope, it becomes a lot more clear. So this side here is the cis face. This side here is the trans. So this is the shipping side, and this is the receiving side. See, there are no big bubbles on this side because the stuff that's coming in hasn't been packaged yet. Okay, it hasn't been packaged, it hasn't been processed, it hasn't been exported. Okay, so it comes in on this side, and all the Golgi really does is put the garbage in a garbage bag. Okay, it packages the wastes. Okay, sometimes it processes it too, but packages it. Okay, you can see on the shipping side or the trans side, there are lots of bubbles, which we call vesicles. Okay, those vesicles are all on their way to the cell membrane. Okay? They contain those waste products and they're taking them to the cell membrane. So on one side of the Golgi, lots of vesicles. On the other side, virtually none. Okay? It wasn't until the electron microscope came along that people figured out exactly what it was the Golgi apparatus was doing. Okay? Because they were actually able to see there's a lot of differences between the two sides here. Okay? So if we're writing down what the Golgi apparatus does, okay, basically it's going to be exporting, packaging, processing wastes. Okay, so controlling the export of waste products, something like that. Okay, questions on the Golgi? Okay. Now, lysosomes present only in animal cells. Okay, and you can kind of see the process here. And this is actually mislabeled. This is actually a peroxisome, and they're produced by the Golgi apparatus, believe it or not. Okay, so they contain this harmful stuff. Okay, that can digest food. And what's food made out of? Well, yeah, the same stuff a cell is made out of, right? So you really don't want the stuff that's in a peroxisome or a lysosome to get out, because its whole job is to break down cellular material. Okay? In fact, it's how an animal cell will destroy itself. Okay? Animal cells have like a self-destruct mechanism. Okay? If they detect that there's something wrong with them, they will rupture their lysosomes, and the lysosomes will destroy the cell and kill it. Okay? The reason an animal cell does that is to protect the rest of the organism. Okay? Most of the time, that machinery works properly. Sometimes it doesn't. If it doesn't, then the cell can become cancerous. Okay? What cancer really is a bunch of your own cells growing and dividing out of control. Okay? The part of the cell, when the cell becomes cancerous, the part of the cell that really is malfunctioning is the part that controls cell division. It constantly signals divide, divide. It's time to divide. Okay? And so they keep dividing, and we get this mass of cells called a tumor. Okay? Now, you might be thinking, okay, well then why is cancer so bad? It's just a lump of cells. Well, that is bad, because when your body starts producing more cells, it'll also start doing things to support and feed those cells. That will be redirecting nutrients, okay? Growing veins and things like capillaries and stuff like that to feed and supply those cells. Well, when you get a big enough mass, you're now depriving the healthy cells in that same area of the needed materials, because you constantly have more and more and more cells that need to be supplied. And they're carrying out a high energy process, division, all the time. So they essentially starve the healthy cells around them and kill them. And that's what can lead to organ failure from a cancerous tumor. Okay, so if you got liver cancer, it's essentially a big tumor stealing everything from all the healthy liver cells. Okay, so lysosomes, their job, digest food. Okay, so this is the peroxisome. Okay, when digestion is occurring, okay, that's a lysosome. Lysosomes are also used to recycle cellular material. If an organelle stops functioning, the cell just eats it. Okay, it's the ultimate recycler. Okay, it's not just going to say, well, that mitochondria is no good, throw it away. Okay, it's not going to put it out of the cell. It'll break it down, okay, and use the raw materials. Okay, the mitochondria is the next one. That's where cellular respiration occurs. That's really what you need to know about it. It does have a special membrane, double layer. It insulates the rest of the cell against the process of cellular respiration, because cellular respiration generates a ton of heat. Hey, how many people have ever had a fever? Okay. When you have a fever, your body is signaling all of your cells to run hotter than normal. Okay? They burn way more energy and they get hot. Okay? Your body's trying to drive your temperature up, because usually a fever will kill the infection. Okay? Because a higher body temperature means the cells that are infecting you are not going to be happy. Neither are your cells, okay? Which is why a high fever for an extended period is bad, but it's your body's way of weakening the infection so your immune system can kill it. Okay? It doesn't always work, but a lot of times it helps. Okay? So that's what the mitochondria basically carries out cellular respiration, and they generate a lot of heat as a result. Okay? Okay, chloroplasts. Only in the plant cell. Their main job, carry out photosynthesis. Really all you need to know about them. I'm not going to ask you about the different parts. They also have a double membrane, because absorbing sunlight can lead to a lot of heat. Okay? So they can end up generating some heat as well. All right, the other organelles of the cell, and hopefully we'll get to all this. Okay, the other organelles of the cell, okay, the cell membrane. I was telling you, it kind of looked like Jell-O salad. Okay, if you look, whoop. Okay, at the cell membrane, this is what it actually looks like. Right? So there's lots of fats. That's the main part of the membrane. But there's also proteins, sugars, glycoproteins, which are like fats and proteins, okay, and sugars and proteins combined into large molecules, big masses, okay, kind of stuck within the membrane of the cell. There are proteins that are designed to help transport material through the membrane, okay. There are some in there that are receptors, so they're triggered by the presence of hormones. Okay, they're a receptor for a certain hormone or something like that, but it's a much busier place than most diagrams lead us to believe. Okay? All right, so main job for that, controls endo and exocytosis. All right, so we got the function right there. Cell wall, this is the cell wall right here. Okay, this green part, that's what the cell wall looks like. It's hollow. Okay, like I was telling you the other day, it's a frame, not a wall. Okay, a wall you can't get through. Okay, but a frame you can walk through. Okay, that's kind of what the cell wall is. Okay, so its job keeps the shape of the cell, supports the cell. Don't pack up. You can pack up when the bell goes. You got a long break. You'll be able to get where you need to go. Okay, your water vacuole. Okay, we'll finish those tomorrow.